Okay, good morning and good afternoon to everyone. And I am Iffah, Syafiqah, Saidatul, Nasuha, Sazwani, and Nur Zulaina, and we are from Group 8. Okay, now I will explain about task 1. Okay, this is the properties of glucose oxidase. It is same with other group, but there is an additional information here. Substrate specificity. It also can oxidize dialdohexose, monodeoxo, D-glucose, and methyl D-glucose at varying rates. This is the similarities and differences of glucoosidase found in living organisms of various family or genus. We compare the glucoosidase in Aspergillus niger and Penicillium aeruginosa. Hello everyone. Okay. Glucose oxidase has been used in many industries, and from biosensor to monitor the blood glucose level to the food preservative and color stabilizer. So there are 6 applications, and the first one is glucose biosensor for diabetes monitoring. Currently, patients who have diabetes will monitor the blood level using a finger prick blood sample, but now we are using a biosensor to detect the fluctuation of blood level. And then there are many more. But many food industries use glucose oxidase, such as in food and beverage additive. The main idea is glucose oxidase will remove the glucose and oxygen, which is it will prolong the shelf life of the food. And as we know, glucose oxidase also yield a product such as hydrogen peroxide, which is hydrogen peroxide is a good antimicrobial, and it has been used in oral hygiene as a bactericide. Okay, thank you. Next, I will continue with task 3. A protocol to measure the enzymatic activity of GOX for different substrates. The reagent needs is here, and the procedure is set the spectrophotometer at 460 nanometer and 25 degree. Why we use this spectrophotometer is because it's easy and simple, sensitive, and requires small sample.
Next, I will explain about the protocol to produce a cell extract from Aspergillus niger. For this, we use solid state fermentation. Solid state fermentation is defined as the fermentation process in which microorganisms grow on solid material without the presence of free liquid. For this procedure, we first isolate the Aspergillus niger from the soils, and then we autoclave separately from the substrate and distilled water. And then we cool and mix the substrate in air media flask, and then we remove the after 66 hours, and then we filter and centrifuge the slurry to get the supernatant. Why we use solid state fermentation? Because it is cost effective, because it is smaller vessel and lower water consumption. It's less effluent release, reduce pollution, and lower energy consumption. Also, it gives rise to better yield of homogeneous spores and support well the growth of fungi and their spore, and the process is simple.
Okay, now task 5. The purification of GOX from Aspergillus niger. So the extract we will centrifuge about 25 minutes to remove debris and ribosome to get a supernatant, and then we will add ammonium sulfate and then to get the precipitate, and then we will redissolve the precipitate and then load to the DEAE cellulose column and then wash with the phosphate buffer, and then we get the pool fractions containing activity. We will chromatography on phosphocellulase, and then the last step is we will gel filtration and get the pure enzyme.
Okay, for task 6 is for one of the method is by constructing enzymatic assay of glucose oxidase. The method is by polyacrylamide electrophoresis patch and followed by Western blot. Western blot is a method used to detect specific proteins in a sample. This method is conducted after electrophoresis. The proteins on the gel is transferred to a nitrocellulose membrane where they are stained with antibodies specific for GOX. When exposed to an appropriate substrate, GOX drives a colorimetric reaction and produces a color. Western blot precisely help in determining the protein is a GOX because it uses antibodies bound specifically to GOX. And for demonstration of no significant contaminants by running on polyacrylamide gel electrophoresis to indicate that only one type of protein present in the sample if one single band produced. The method is by agarose gel electrophoresis. No band form means that there's no contaminant from DNA.
Okay, this is 3D structure of GOX. Okay, I was using Uniprot to get the file for amino acid sequence. So first, you have to find all the organisms that have glucose oxidase. Copy the amino acid sequence and to the text document. Okay, this is the process, and then paste all the sequences in the cluster W2, where these tools will help you to align multiple sequence, which is then we can identify which one was the active site. Okay. So how we determine the active site was there are lots of sequence. We have to look for the stars that located closely each other, and the most higher star that is the active site.
So the last part of this presentation is on task 8. Generally, there are several techniques that you can use to determine the protein structure. For example, circular dichroism, Fourier transform infrared, fluorescence spectroscopy, and NMR and X-ray crystallography being the famous ones because they are capable of giving structural information on protein at atomic level of resolution. But we choose to nail the task 8 using a technique called circular dichroism. So this is how the instrument for circular dichroism looks like. Circular dichroism is the difference in the absorption of left-handed circularly polarized light and right-handed circularly polarized light, and occurs when a molecule contains one or more chiral chromophores. Why we choose circular dichroisms? As you can know, for X-ray crystallography, it requires a suitably refracting crystals of protein, and as for NMR, it requires a high concentration of proteins and it is limited to relatively small proteins. And we know that the molecular mass of GOX is pretty large, so we couldn't use NMR. And the good thing about circular dichroism is that it is much less demanding technique, both in terms of sample and the time requirements. And CD also could be used to assess the structure and stability of a protein under conditions used for NMR, and it can use to explore structure under conditions close to those used for crystallizations. So that is the end of this presentation today.